Good morning, everyone, and welcome to day 2 of our Midwest Vascular meeting. We hope you all enjoyed the welcome reception last night. We want to thank all of our industry partners once again for their support and encourage you all to speak with as many exhibit reps as possible throughout the day. As a reminder to all presenters, please verbally disclose any relationship with industry prior to the start of your presentation. We also want to ask everyone to ensure that their cell phones and pagers are silenced. Thank you. And with that, we will welcome our first presenter to the podium, who is Dr. Christopher Odu, to speak to us on Diminished JARID1C activity alters diabetic wound plasmacytoid dendritic cells and impairs tissue repair.
Good morning. Good morning, everyone. Thank you for that introduction and the privilege of the podium. Our lab has nothing to disclose. So this audience is very familiar with the perils of non-healing diabetic wounds and what eventually can happen with resulting in amputation and even other sequelae. And we also know that a lot of these things are due to inflammation, just overriding inflammation, which prevents these wounds from going through the normal healing cascade as shown in these different colored histograms that you see there. Now, we also know that in diabetic wounds, they all have just multiple different cell types that are in there. And our lab has been specifically focused in looking at macrophages. But of recent, we were more interested in trying to understand what other types of cells within the diabetic wound contribute to inflammation and poor wound healing. And one of the cells that got our attention were T cells, because T cells have memory, and T cells are involved in promoting chronic inflammation. So to get at that, we started by looking at human wound T cells, diabetic and non-diabetic, and looked at what signature they had. And what we realized was that diabetic wound T cells had a higher level of Th17 inflammatory T cells. And that was also true in our mouse model, which when we created diabetic mice by feeding them a high-fat diet and then looked at their wounds, and what we noticed was that they also have an elevated level of Th17 T cells, and they had elevated levels of IL-17, which is an inflammatory cytokine. So we began to wonder what types of cells prime these T cells. We know macrophages can do it, but we were interested in looking at other types of cells within the wound milieu. And what we did was we realized that when we looked at the chronic inflammatory literature, plasmacytoid dendritic cells really stood out as first responder antigen-presenting cells that really link the innate and adaptive immune cell response. In addition, they're known to produce a lot of type 1 interferons. So I've highlighted kind of the relationship that we postulated here, that PDCs are perhaps what are involved in priming these T cells towards an inflammatory phenotype. So we therefore hypothesized that diabetic wound-derived PDCs are physiologically altered and contribute to chronic T cell-mediated inflammation. So what I'm showing you here is a diagram that shows the different types of T cells and what it takes to become a Th17 T cell from a naive T cell, which is seen in the purple to the left of that screen there. And what it takes is elevated levels of TGF-beta and IL-6 in particular in order to create a Th17 phenotype. So we took our diabetic wound PDCs and asked the question, do they make a lot of IL-6 and TGF-beta? And what we realized is that they do. You can see here, the DIO stands for diet-induced obesity, and what that shows is that they have elevated levels at the protein level of IL-6 and TGF-beta. So we then took those same cells and incubated them with naive T cells, because I wanted to ask the question, when I incubate these with T cells, will I still get a Th17 phenotype? So what I'm showing you here is kind of the workflow of what we did. Took the PDCs from diabetic mice, incubated them with naive T cells from normal mice, incubated for a few days, and then did flow cytometry. And what we realized was that those T cells that were incubated with diabetic PDCs had increased levels of IL-17, and they had an increased signature for Th17, which is denoted by the ROR gamma T there. So this was exciting to us because we're starting to piece the puzzle together that these PDCs produce these cytokines and skew T cells towards this type of phenotype. But as you all know, in science we have to prove it twice. And so what I did was we repeated this experiment, but this time took T cells from normal mice that lacked the TGF-beta receptor and or included IL-6 antagonism to see if that actually would drop down the levels of IL-17. And indeed, they did. So if you look at the graph on the right, what you see is in gray, to the far left of each graph, you have the normal naive T cells with diabetic wound PDCs. And when you treat with an IL-6 inhibitor, you knock that down significantly for IL-17 and the Th17 signature. And in addition, when you look at the ALK5 or the TGF-beta receptor knockout mice, those are down as well. But importantly, when you do both, you have the lowest levels of IL-17 and ROR gamma T. So together, this suggests that diabetic wound PDCs are hyperproducing these cytokines that result in these inflammatory T cells. So then the question becomes, why is this occurring? And the answer lies in epigenetics. So epigenetics is that field of study that involves non-hereditary genetic changes that can influence the phenotypes that we see. And the one that I want to point out to us is this one in particular, where the trimethylation of lysine 4 on histone 3, or this H3K4ME3, which if increased, leads to increased gene expression and increased inflammation. So we started by looking at, pulling down the H3K4 mark in chromatin, and what we noticed was that mark was increased at different promoters, at the IL-6 promoter and the TGF-beta promoter in the diabetic state. And so there's two ways that can happen. It either is because you have too much methylation going on, or you have too little demethylation going on. And so to get at that, we performed what's called a histone array to figure out what enzyme is responsible for this. And what we determined, and I acknowledge that this is difficult to see, but what we determined is that JARID1C, or lysine demethylase 5C, was actually diminished or decreased in the diabetic state. And indeed, when we looked at the mRNA levels of this, we saw that same trend, and we saw that same trend at the chromatin level as well. JARID1C in diabetes is really low. So we did the reverse experiment where I took those wound PDCs, treated with recombinant JARID1C, and then looked at what and then treated with T cells, and noticed that IL-17 was significantly decreased when you gave back recombinant JARID1C. So together, this suggests that this epigenetic enzyme is involved in this process. So finally, we wanted to ask what controls JARID1C expression in diabetic PDCs. Well, what we found was, what we postulated was that it was due to type 1 interferons. And so when we took these PDCs and treated with interferon beta, what we realized was that we had an increase in JARID1C and a decrease in the K4 mark at the chromatin level in the diabetic state. Similarly, when we looked at mice that lacked the interferon receptor, they essentially exhibited the same things that we saw in the diabetic state, where you have increased IL-6, decreased JARID1C, and increased K4 mark. And to get at the signaling cascade, we did a quick experiment to look at when we inhibited with tofacitinib, which inhibits JAK1/3 signaling, and a TYK2 inhibitor, and noticed that led to increase in IL-6 in the presence of interferon. So I know I said earlier that PDCs are known for producing type 1 interferons. So am I double speaking here? Well, it turns out that in diabetic wounds, there's a decreased levels of type 1 interferons, which is in part responsible for why these wounds don't heal. And now we realize that the question is then, so what about these PDCs? If they make a lot of interferons, why aren't we seeing this change? The question, there are two reasons. One, we realized that there are fewer diabetic PDCs that hone to the wounds after an insult. If you look at the bottom, at this graph, you see that the dark circled mark shows the diabetic curve, and there's an order of magnitude less compared to normal diet. And then these PDCs also make less type 1 interferons. So this was an interesting and exciting study that really has opened the door for multiple explanations or trying to figure out why this is, which unfortunately I don't have time to get into. So just to summarize, we believe that in the normal state, type 1 interferons, there are a lot of type 1 interferons, which leads to an increase in JARID1C, which then leads to a decrease in K4 mark, decrease in IL-6, and improved wound healing. The opposite is true in the diabetic state, where there's less type 1 interferons, there's less JARID1C, and there's an increase in IL-6, which leads to increase in inflammatory T cells ultimately. So this is exciting because it points out that there are multiple places now that we can do drug development and drug design and target for a new mechanism for wound healing. And with that, I'd like to say thank you to my lab and all my mentors and our funding sources, and to you for listening to Basic Science at 8 a.m. in the morning.
Thank you. This paper is now up for discussion, and I have a question for you.
Sure.
When you took the mice and you had the wounds that either had the ability to have the dendritic cells or not, did you continue to go on, instead of just harvesting those, did you see what was the wound healing difference? Was there any time to wound healing difference, anything that you could see on a macroscopic basis?
So if I understand, you're asking, when I looked at the PDC, I guess when I replenished the PDCs, actually, yes, it turns out that if you knock down PDCs in diabetic mice, you actually get improved wound healing. And I didn't show that data here, which was really interesting to us because it suggests that by decreasing the amount of PDCs that are there, you're also decreasing the inflammatory cascade, which was interesting to us.
Was there a time frame difference, or what were the kind of the endpoints that you saw?
Yeah, so by day 3, actually by day 5, day 3 to 5, these wounds were drastically healed and smaller in the treated mice versus the non-treated mice. So that suggests that PDCs are a viable target for us to eventually look at for wound healing. Yeah.
Thank you.
Yeah, you're welcome. Thanks.